So I grew up in New York, Long Island, New York, and I was raised in Suffolk County, for those of you who know New York, so out in the boonies, and had a great childhood, really enjoyed the outdoors. Used to sail a lot in Long Island Sound, and you know, something that always lifted my spirits was being out in nature and really enjoying everything from literally hiking to the trees around me and so on. Those are things that always resonated really well with me. And once I finished high school, I got in my car and headed north to University of Albany to start my undergraduate career, and on we went from there. So who knows where you really get these inspirations, but mine has always been looking around at nature and just trying to understand the fascination you see around you all the time. So those of you who don't know me well, my one and only religion in life is trees, and I say that with seriousness, right? And if you've never spent time just sitting outside, either wandering at trees or under a tree, you know, give it some time. If you think about what trees accomplish in life, right? They usually live way longer than humans, particularly if you go with the really ancient trees like the giant sequoias and the redwoods. They also are remarkable because they connect the earth to the sky and everything in between. And they're living beings, right? And they connect and communicate with chemicals and other means, and probably things we don't understand. And yet they're invigorating. You know, people talk about, you know, walking in trees and you feel different, and who knows about that. But I was always fascinated by how something so simple, right? You think about what makes up a tree. It's basically sugar, some salts, a few other odds and ends, and a few enzymes to make something like cellulose, right? Which is just link sugars together, lignin, which is just a couple of polyphenols, and all the other polymers in there, and yet you have this amazing living structure. So that's where my inspiration has always come from, and that's even what led me in my PhD work to study lignin biosynthesis. That was my biochemistry-related PhD thesis, was how is lignin that makes up trees synthesized. So it's a lot of enzymology, a lot of chemistry, and a lot of biology. So that's persisted to today from the inspiration from my own lab, which has always been bio-inspired materials from the very beginning. Way too long now, right? Decades, but whether it's polysaccharides, whether it's silk proteins, whether it's how tissues assemble, whether it's structural hierarchy, it's always about how nature has simplified the building blocks, but yet the chemistry in those building blocks drives the assembly to make these really remarkable structural and then functional materials. And so our job is to kind of reverse engineer that, figure out how it assembles, and then try and do it in a different way to make something useful, whether it's for medicine or food or something else in between. Well, look, the tools of technology, I'm all in. You know, give me any tool, I'm going to use it. You know, people can decide whether it's right for them or not. That's an individual choice. But in terms of doing science and engineering and trying to understand first and then make things that could make the environment better, people better, whatever, you know, I'm all in. And I think ultimately genetic engineering, as I'll just call it, the next level of, whether you want to call it evolution or whether you want to call it something else, but it's a natural progression from, you know, mixing genes that happen since, you know, living things started on Earth. It's just accelerating the process. So to me, I have no problem with it at all. We continually use it in our own lab, whether it's to re-engineer materials in new ways. We do this with cells to re-edit cells and change their properties. And, you know, if you're going to use something for medical need, medical devices, and people need it to survive, my guess is 99% of the time they'll probably accept it. If they're going to use it in a more optional mode, whether it's something they could wear and it came from genetics or something they could eat, it's not going to be 99%, and that's fine. You know, there's plenty of people on the planet to make those choices. Yeah, it's sort of the polymer that never goes away. You know, it's like 35 years of studying silk. So whoever thought you would study one polymer for 35 years and still making new discoveries, right? So it started as very simple. One of my colleagues handed me a paper, and the paper was written by a faculty member at North Carolina State University I think he was in the textile engineering department. His name is Robert Work. He's long ago passed away. And this is full embarrassment, so it doesn't bother me, right? So I read the paper, and it's mostly just a set of tables. And the tables are all about the mechanical properties of spider silks. And so I'm like, this is like, and the properties were remarkable. First, let's remember that. And so I was intrigued by it. Number one, that you could find these orb webs, spider webs in nature, and you have these remarkable properties. So the first thing is the science brain goes off, and it's like, I got to understand how this is made, right? Who knows why that happens, but that's always the way. It's a material from nature, you want to understand it. So started digging into the literature to find that out, realized it was very little known, and that's what got us started. The second reason I got started on spider silks from that same paper is when I read the acknowledgments of the paper. Robert Work acknowledged the National Science Foundations for supporting his research to travel all around the world to collect these silks. And literally, he had to travel all around the world. So the light bulb went off, right? First of all, I get to do the science I love. And second of all, somebody's going to pay me to travel the world. It's like, sign me up. And that's really where it started. And the amazing thing is it's all come true. Not just travel the world, but travel the world probably 20 times. Meet people everywhere and inspire hopefully a whole family of silk researchers. So, that's how I got started. And we continue to work on it because despite it being a polymer, it's not like any other polymer that we or anybody's ever seen. And my favorite thing is when you have somebody who studied, for example, synthetic polymers, comes to the lab and has their training in it and all that, and I love the fact they come in bold and confident and all that, and it only takes about a month before they realize they're starting over, right? Because it's a whole nother ballgame when you deal with something like silk. It's just not your normal material, which we love, but it makes it more complicated to deal with. So first of all, if you think about it, it's you never need chemical cross-linking or photo cross-linking, and you get these remarkable properties. So that's a really important starting point. So you don't have to add all this other stuff in to make something that can be robust for all kinds of medical devices and related applications. The second is it's a really high molecular weight protein, pure protein, but it's essentially nature's equivalent of a block copolymer. And so, we think we're so clever, right? Chemistry-wise, we build all these block copolymers. Nature designed this eons ago, right? And we still haven't emulated this protein in the lab. So for example, when you look at a spider silk or silkworm silk, you have all these domains. For a really strong silk, that means those domains are mostly hydrophobic amino acids. So like 80% glycine, alanine, methyl groups and so on. So it's incredibly hydrophobic, but there's just the right balance of hydrophilic domains to keep things soluble in water in what we call a metastable state. It's ready to go into a solid state. And that makes the challenge of processing, inherent because you do the wrong thing. You blow on it, you trigger it, and it's going to convert to an assembled state. So you have to work with the processing. That's number one. Number two, if you look at those domains in the silk, the hydrophilic domains are highly, highly conserved, right? So that means you have just the right amount of charged amino acids to get this balance in solubility in order to have this sort of precursor ready to spin into a fiber. But if you look at the hydrophobic domains, they're all different sizes. And that's what we haven't emulated in synthetic block copolymers. And we believe, our hypothesis, not fully proven, is that keeps the silk, when it assembles, from growing crystals that are too large. They're self-limiting. So they can only grow very, very small crystals, beta sheets, when the silk assembles. And why is that important? When you look at the silks, it's remarkable. It's the toughest stuff, but it's really, really strong at the same time. And that's because you have like 50% is crystalline, but they're teeny tiny, nanometer scale crystals dispersed throughout the matrix. And so no one's emulated that synthetically. So that's where you get these remarkable properties. I could keep going. For example, you put silk in the body, it's fully degradable, but it degrades very, very slowly, enzymatically, right? Which is completely different than all the degradable polyesters that you see. And the products of degradation are amino acids, which is awesome, right? Those are all compatible, normal stuff. And over the years, we've learned to morph it into, a gel, a new kind of fiber, a sheet. More recently, we do plastic parts out of silk with thermoplastic processing. So, you can do all those things. So versatility is a big one. Everything start to finish is in water, pure protein, degradable, compatible, and so on. And then the last thing I'll mention, because it's becoming sort of well-traveled now with some of the startups and the spin-offs, but silk, because of that amphiphilicity, right, hydrophilic balance, you can put bioactive compounds in there, enzymes, drugs, even cells, depending on what you're doing, and they stay remarkably stable. So you can make a silk part as long as it's not immersed in water, stick it on a shelf, the enzymes in there stay stable as long as it's there. And because they're not active at that point, but they're not deactivated either. And then when you immerse it in water, water will activate the enzymes. So again, those are some examples of why it's, at least we think, pretty remarkable material. So it depends on what your goal for your research is, right? If your goal is to do fundamental research, then we use genetics. And so we focus more on spider silks because there's a diverse array of spider silks with different properties. And so those we clone, design genes, clone, express and study structure, function, and learn from there. If your goal is to make medical devices, the only game in town is really silkworm silk because you can buy it by the truckload from the textile world, and then you can use that, purify it, and clean it up, and it's very relatively inexpensive by standards of medical materials. So it's simply a supply and demand issue at this point. There are maybe a half a dozen companies around the world trying to commercialize genetically engineered spider silks, but the cost is still way too high and the supplies are not there yet. So for anyone that works on tissues, every matrix in a tissue is going to be different depending on the particular tissue. So as a starting point, you need materials that are cell compatible, and I would just say versatile, and I mean versatile in morphological features, chemistry, mechanics, and everything else, right? So again, coming back to all those virtues of silk, it's a slam dunk, right? And I don't know of anything else that has the versatility for all these different tissues. So whether we're growing an injectable, cell-laden injectable gel to fill a defect in the body, or whether it's a bone tissue where you need really stiffer, stronger material, or brain tissue, silk can fill all those roles. And the other thing, if you, like our lab, when we grow human tissues in the lab, one of our mandates is it has to be physiologically relevant. This is just something that the group kind of abides by because of my own drive here, right? One has to be physiologically relevant for the human condition. And number two, it has to be what I call sustainable. And I don't mean environmentally sustainable. I mean, it has to be there and you could study it for a long period of time to really understand not just acute effects in human tissues in the lab, but also chronic effects. And a great example for one of the things we spend a lot of time on is the human brain, right? Most of the challenges in studying the human brain aren't, what happens if you hit the brain over the head like a TBI injury, right? The challenges are, how do these neurodegenerative diseases develop? And they're not going to happen in a day or a week. You've got to be able to grow these things for weeks and months, if not years, to really let the disease develop and then look at intervention. So that's where silk comes in as well. It just doesn't degrade very quickly in vitro unless you make it degrade. And so that gives you that stable structure that cells enjoy. And for some of those reasons, that's why it's really proved to be versatile for these kinds of repairs. Yeah, I mean, usually it's not a problem because of contact inhibition of the cells, right? So as long as you provide the surface area, the cells will eventually bump into each other and their own programs will go into place to control the growth. The only time you don't control the growth by design is when you're using cancerous cells. So if we study glioblastoma, you want them to keep growing, and that's what we see in vitro and what we've published on this. So So again, the cells are pretty self-regulatory. The silks, depending on what they are, porous foams or whatever, give confined space, and they seem to do really well. Yeah, so again, that's another advantage of silk because it's basically an insulator. It's not a conducting material. So you can decide if you want to add in electronics or not to the systems, which we've done. But some of the work has just been inherent in understanding cell electronics, if you will, right? And this is all work we've done in collaboration with my colleague at Tufts, Mike Levin, where we look at a lot of voltage-gated channels, and essentially we think of a cell as a battery, if you will, right? And so there's a differential in terms of charge across the membrane, and so this voltage difference is important in all cells and all tissues. And so when you form a tissue, those cells have to line up and connect and communicate, and that creates now a tissue level differential in terms of those electronics. And that goes on and on. So, you know this from the literature. If you have skin, you've got a certain sort of membrane potential across your skin, and these fields exist throughout your body. If you damage or cut your skin, all of a sudden those flow fields change. And so that's what we try and emulate in some of our 3D tissues to understand those electric fields self-driven. So these aren't externally applied fields, but how do they change, and then how can we reprogram those cells to repair in ways that that people don't normally think about. And we reprogram cells with changes in those channels as well, either overproduction or inhibit the small molecules to look at how that affects the growth. And a good example is the early work we published now. It's getting out there 8 years ago or so. You could take a human stem cell and do nothing other than change the voltage across the membrane of those cells. And you can cause it to differentiate, you can cause it to trans-differentiate, you can cause it to stay a stem cell, and so on. And so it's just a whole nother repertoire you can use to drive cell behavior and ultimately tissue behavior. And Mike and I now are working on things like limb regeneration, which again is the holy grail, right? If you are a human and have lost a digit or a limb, what can you learn from these kind of voltage changes that you might be able to exploit in a new way to enhance complex tissue regrowth. So that's part of the ongoing work. So I have to give some context to this so I don't look like a total idiot, right? So, so because we have a long history of tissue engineering in the lab, and we've been fortunate, we're now in our 18th year of support from the NIH through what's called a P41, a resource center on tissue engineering. And we had it for 15 years led by Tufts, and now my partner at Columbia leads it in the new iteration, which is great. And so we had this history of tissue engineering, and then I had two things happen back about 10 years ago. I had a DARPA-funded project, and DARPA put out a call for proposals, and those of you who work with DARPA, DARPA doesn't want to fund things that somebody else would fund, right? It has to be something that's, kind of out-of-the-box ideas. And so they were looking for new ideas to make the next generation of robots. I'm not a robotics lab, right? But we know how to grow tissues, and I have a colleague at Tufts, Barry Trimmer, in biology, who has studied caterpillars. So we got together and we said, well, why can't we propose to use tissue engineering to grow robots, right? You could grow muscles and make those as the actuators. You could grow the skin. These are all things we've been doing already. And our idea was even taken to the next level. We could actually use fat as the energy store, like to drive the cells. And so that was what we wrote up. And Barry studies insects, so we said, well, if you're going to grow these robots and put them out there in the environment, they better be hardy, right? They can't be mammalian cell driven, because mammalian cells need such tight control, care, and whatever, that they'll never survive. So we wanted something that doesn't care what you do to them, right? And insects you find everywhere on the planet, doesn't matter where. So insects are pretty hardy. And so that's what we did. We picked a particular caterpillar that he was studying. We took my engineering tools from the lab, and we proposed to DARPA. We were going to grow robots. We were going to build them. We were going to grow them. They funded it, right, for 3 years. And not only were we surprised they funded it, we were happy they did, right? So we went to work on growing muscle and growing fat and all these things, that was 8 to 10 years ago, a bit crazy, if you will. And what happened is, the really nice thing is some of the students that came to the lab to do those studies, they were undergrads, grads, and postdocs. They all really got intrigued by the idea, using cells in ways other than just straight medicine. These are different ideas. And a couple of them came to the lab and said, well, can we grow food this way? And I was like, well, I don't know about food, but let's work on robots. So that's where it started. And I will say almost all those students from back in those projects are now leading their own labs or CEOs of companies in the cellular agriculture space, because that's where they got started. So it's pretty cool to see how they've evolved. And then 7 years ago, after a bit of a lull when that project finished, a prospective PhD student showed up at my door, and she said, Dave, I want to come to your lab and I want to grow food. And it's been a few years since the DARPA project. I said, well, we're not working on food. So I said, this isn't the lab for you. And so she said, but you do tissue engineering, and that's what I want to learn to grow food. And I said, well, wasn't so sure about it. So I sent her away, and she's a very persistent student. Her name is Natalie Rubio. She came back again, and she said, look, I have a fellowship from a nonprofit called New Harvest. So I just want you to let me get in the lab. And this is how I don't know anything I'm doing, guys. I'm just telling you my history, but in fact, I was sure that Natalie would never make it in the lab. I told her this too. We were very open about these things. I said, you're going to get started, but I think you'll be bored in the lab. This isn't really my sense. She's been one of my best students ever. She just graduated with a PhD in December. She's now heading up on one of the cell ag startup space areas in bioreactor design for the next generation. But she was my first full-time PhD student growing food, by her demand, not mine, right? And that led to a whole bunch of other students coming in because she's got this great personality as well. So they all came to the lab. And now I think we have 20, as of September, we have about 20 graduate students and postdocs dedicated 100% to cell ag. Almost half my group now is working on this. And equal number of undergrads, equal number of master's students. But it's booming. And why is it booming? It's booming because we need options to feed the planet. We need options because of sustainability. We need options because of food equity. And from the medical side, we need healthier food. If you want to really know where your food comes from, look at the videos and read, and you'll be, horrified. Doesn't mean you can't eat the stuff. You just have to be aware of, how much antibiotic use, how much hormone use. It's pretty depressing, And so we're out to build a science foundation for the field. The field grew more out of private investment in startups in the space, which is great, but there's no really fundamental science that's been published in the field. So that was our goal, and that's still our goal. And so we've published now on growing bovine muscle cells that we do biopsies at our vet school at Tufts. We grow them out in the lab, we study them, and now we're just reporting out the first immortalized cow muscle cells, right? That'll be great for everybody in the field to be able to use. We've immortalized fish cells now, and you need these cells to really grow the incredible volumes of food you're going to grow eventually. So that's how we get started. And sure, back to our core, principles. It's all about the biomaterials that you need to host the cells. Now, these are different design rules, right? These are not silk-based because they have to be edible, digestible, maybe nutritious, and so on. Silk just doesn't degrade very fast in the gut. So maybe not the best option. And then the overriding thing when you grow food, unlike medicine, is cost, right? So you have to find things that are available in large scale, cost almost nothing, that you can use for food. Unlike medicine, where you can use anything pretty much. As long as you can make it work well, people will use it. So very different design goals, but the ultimate need is the same. Getting cells to do what you want at a scale you need to make the right mechanical and other features that you need for the given task. Well, we're still figuring it out. I mean, it's ongoing work. But yeah, you work with muscle cells because that's where a lot of the texture of meat comes from, right? These are things I never used to think about, just so you understand, right? But when you talk about the flavor and taste of meat, it comes from the fat, for the most part. And so we spent a lot of time on fat cells. So again, whether it's porcine fat cells from pigs or bovine or fish, Same idea. How do you isolate those cells? And these all start with adipose-derived stem cells, just like muscle or muscle satellite stem cells. And then you have to figure out how to cultivate them. And the first thing you learn very quickly, the students learn, is you can't go to the literature and find, okay, what are the best markers for these cells? They're all out there for human cells, but there's almost nothing for every animal cell you want to grow, isolated grow, to know, do you have the right cell? Do you have antibodies? Do you have primers for PCR? Everything's different. And so it takes a lot longer to characterize these cells, figure out how to grow them well, and so on. But that's what we and others are doing now. And then for the fat cell, one of the real nice features for fat is when you're growing those in culture for food, you can feed those cells directly, right? If you think about an animal growing that you're going to feed things to change the fat content, you have to go through all these different, I'll call it metabolic barriers, what's in the stomach, how to get it through the intestine, all the things that go on. So controlling the composition of the fat is very, very difficult. When you're growing cells in culture, you have direct access. So it's amazing. You can manipulate the fatty acid composition of those cells. So start to think about healthier eating, right? Right away, that fat becomes healthier, and so on. So I think there's a lot to be gained from doing that. On the other side, if you go back to the muscle cells, you can now, and we've published this already, you can take, for example, cow muscle cells, and we could use synthetic biology again to introduce pathways that don't exist in these cells. So the one we've published so far is putting in something like beta-carotene that normally you get from carrots. So now you think of a hybrid. You have a muscle cell from a cow combined with carrots all in one, right? So it's one-stop shopping. But the point is healthier cells, and think about how much that also buys you. It buys you, in principle, higher antioxidant content of those cells, which in principle means more stable muscle cells on the shelf before you buy it, and it also means healthier when you eat it because of that antioxidant level. So, these are the kinds of things we're looking at. I think it's only the beginning, and I think there's still huge challenges ahead if you look at the scale to grow food in the future. But we have no choice. If you look at the population, people project now will be at 10 billion by just after 2050. We're at like 8 point something right now. We're running out of land. The land consumption to grow cattle or grow chickens is just not tenable, right? Not to mention the conditions they're grown in, and as I said before, the overuse of drugs just to keep the animals healthy enough. So we have to do something. Right? This is one option going forward. We have talked a lot about it. We've generally not bothered with brown fat because when I think about it, because it's metabolically more active, that's actually a detriment when it comes to the food, right? Because it's going to be self-consumed or not available as readily. I may be wrong, but that's kind of our attitude. Whereas the white fat is perfectly fine, particularly when you start to make the composition what you want, which is what we're trying to program in. So yeah, I think people will look at all of it, and I think there's value probably somewhere there, but we haven't really explored it very much yet. I think you're right. Fat is many different things, right? And it boils down to the composition, whether it's the fatty acid profiles, phospholipids, other flavors that may be in there, right? So we don't know. But, there's a couple of holy grails here. One, those of you who work with stem cells, one of the holy grail has always been like a universal cell, right? So you don't have to worry about whether it came from you, or you. We could all use the same cell to repair tissues, help a patient, whatever it is. Doesn't exist yet. People are working on that with CRISPR and other things, right? Same applies to food. If you follow at least the food literature, the argument is if you have fat from one animal versus another, it's really the composition of the fatty acids and the phospholipids again that drive that flavor and taste. And so in principle, with what we're doing, we're not there, but in principle, you could have just a generic fat cell and then simply control what the composition of that cell is. And so you could have somebody's going to Amazon for their career, right? You could sell the Amazon of fat cells, right? And you could order whatever. I want fat cells to taste like chicken, or I want them to taste like ham, or whatever it is, and you've already programmed those cells to have the right composition. And so, those are things we spend a lot of time on, we think are very viable, but it's still early days to get there. Yeah, sure. So we started, oh my God, it really dates myself. We started studying bone through tissue engineering probably close to 20 odd years ago. And back in the early 2000s, we published a whole series of papers. And the whole focus back then is back to silk, right? First of all, knowing silk had really nice mechanical properties was about understanding how to engineer the silk with the right morphology and all the things we talked about, chemistry and so on, to get bone cells to really want to be in there and create their own bone matrix, but keep the mechanics up, right? And so we learned a lot of things back then. One is, yes, you can take silk and make a porous scaffold to grow bone in that, and that we showed many different ways, whether it was load-bearing bones, whether it was cranial bones, and so on. And one thing we learned that we published, which I'm still amazed at, is the most highly cited paper from our group in all my years, was a simple paper that we published in Biomaterials about how important pore size was for cell function in those scaffolds. And we published, basically, you needed to be at least 300 microns and up if you're going to have a viable, non-necrotic core over time. And so back then we did all that work. We did animal studies, both with clinical colleagues in Boston, at Children's and elsewhere, at Beth Israel, and then we did stuff at the vet school and our dental school to show these things worked well. And so we had a whole family of scaffolding that worked well with bone cells or without, and on and on. What we learned after that was no one cared. Okay, and it's not that no one cared, but there were so many other, I guess, simple, cures out there, whether it was synthetic materials or demineralized bone. So there wasn't enough, what I would call, novelty to what we made, that at least it was ready for prime time. And that's fine, that happens, right? And so we kind of put that away as we started all our tissue engineering with bone and cartilage as the kind of obvious starting points. And that's where we went off into other more exotic tissues, if you will. And those included skin, full thickness skin, and the intestine and the brain. But I would say back now, maybe 5, 6, 7 years ago, we started to take a new look at bone, and not so much for the scaffolding yet, more for degradable orthopedic devices. So screws and plates, particularly for children. So this again was done with colleagues at Children's and so on, where we said, right now the only option in town were the PLA screws. These are very tiny if you've ever worked with them. They're very tiny screws for repairing. And so we went about making all silk-based, very dense parts, medical devices out of silk, plates and screws, and we showed they actually work not so much in the heavy load bearing, but you could attach ligaments to bone and fix pieces of bone that weren't load bearing, and that was great. But it turned out that for children they degraded too slowly in vivo. So what we designed worked, but it didn't work well enough. It would probably take years for those devices to fully degrade, and kids' bones grow really fast. So the mandate was, Dave, how can the lab make these degrade in 3 months or 6 months so they get out of the way faster? And so we went back to the drawing board and we designed a whole new technology that we only just first published a couple of years ago, which is thermoplastic molding of silk. And this is a crazy idea because if you look at the history of silk, from the discoveries of silk structure from Linus Pauling back in the 1950s to when we published our paper in 2020, the idea that you could thermally melt silk and have it fill a mold under pressure was heresy, right? Because as a protein and as a structural protein, whether it's silk or something like cellulose as a structural polysaccharide, the density of hydrogen bonding is so high that they always degrade, the materials degrade before they melt. And so this was a real challenge. So long story short, we solved that problem, and so we published how you could thermally plastic process silk, and you could literally, almost like a waffle iron, pop out silk parts, right? So there's your screws and plates again. But I bring it up because now you could do something you couldn't do before. And that is, and again, this is a completely idiotic idea that worked. okay, to take the enzymes, proteases, that will digest the material and put it into the process. So here you're going to degrade your material as you're making it. If you do it right, you can do it, and you can then pressure your parts. Keep in mind, we're talking 125 degrees C, 145 degrees C, and out pops a plate of silk that might look like this, where the enzymes that are going to degrade the part are inside, but they're non-active if you do it properly. And that can sit on a shelf again for years, and it'll be fine. As soon as you put that in the animal, now water creeps in and activates those proteases. So we can tighter in the amount of enzyme, and now the part will degrade as quickly or as slowly as you want. And that opens up the door for all kinds of bioactive medical devices where the drugs, the enzymes, the antibodies, whatever, are completely stable in the plastic part. Keep in mind, it survived processing at high temperature, and yet antibiotics, enzymes, other things are perfectly fine, and on we go. So that's the newer technology for orthopedic repairs, and now we're trying to expand that with the 3D printing, as well as these dense parts to build, replacement parts for broken bones and so on. And that's ongoing work in the team. It's always going to be balance, as you know, in mechanics versus cell compatibility, making sure cells get in there. But you could design those systems now with a lot of really tight control. And one of the things we've been after, along with Vincent Fitzpatrick, who you've interviewed, has been not only to grow the bone with the right mechanics, but also to make sure you provide vascular conduits and innervation as part of the repair. So those are things we're working on. So multifunctional bone systems as replacement parts are growing in place depending on the goal. So these are things that still have a lot of work to do, but I think there's a lot of promise. So again, over the years we've published a number of papers on cartilage. I would say all great in vitro, but no real success in vivo. So whether we've used silk gels and very loose silk sponges, or whether we've worked with collaborators at Columbia, for example, on using alginates in combination, we can get cartilage to grow. We can do all that pretty well, but it's always the interfaces when you go in vivo, as you probably know. So we spent a lot of time on osteochondral interfaces, building multi-level scaffolds to do this, and some of that seems to work pretty well. And my colleague at Columbia, Gordana Vunjak-Novakovic, is continuing that into large animal studies and so on. So I think there's still a lot of promise, but the challenge early days was to just grow cartilage alone was doomed to failure because the metabolic activity is so low and the interface to the underlying bone is very difficult to get it to take. Yeah, back when we did a lot of that work, again, this 20 years ago, we didn't spend a lot of time on those particular features. The challenge, as you know, was always, the biology, juxtaposition to the mechanics, and then, like I said, the interfaces, and difficult problem to solve. So even if it histologically looked pretty good, mechanically it would fail, or it didn't have the right substructures and so on. So I think it's a challenge, and it's a fundamental challenge in tissue engineering. And excuse me, when I teach this, we spend time on it, which is, how do you know when you're done? And I mean that when you're growing a tissue in vitro, you have to pick the right time to implant in vivo. And I think every tissue will be different there. And cartilage, to me, is one of the biggest challenges of all, right? Because as it grows, it's vascularized, highly metabolically active, lots of cells. And then as it becomes mature, there's none of that. And so where is the right sweet spot to get the right level grown in vitro to implant so it's going to take, and then let the rest take care of itself? And I don't think anybody's really solved that problem today. No, it's hard for me to know if they're completely different than what people face today, but I think maybe just at a different scale, perhaps. So, back in the day when I started, you still faced the same challenges, right? You had to get your lab started. You had to find funding from somebody willing to read your proposals. You had to find good students to be the catalyst for the lab. And you had to figure out how to teach. And at least in the day when I went to school, no one told me how to teach. In fact, I didn't think I liked to teach, right? And so you learned on the job, right? And you go from being an independent scientist in the lab, and many of us, I don't know about everyone here, but certainly, you know, I started out as an introvert, so the lab was my safe place, right? I loved being in the lab. No one could bother me. I could do my thing. I could work at my own pace. You know, I was driven, like many of us who go into this, into being to teaching and being professors. But you know, when you start your own lab, everything changes, right? You're no longer isolated and doing your own thing. You know, there's many people depending on you, and all of a sudden you have to turn that switch. It doesn't happen overnight. You make mistakes along the way, and you hopefully learn from those mistakes. And you know, it's important. You have to find mentors who are willing to take the time and guide you in the process. But there are many, many good people out there willing to do that. So, you know, those challenges are really pretty much the same today. You still have to raise money. You still have to write grants. You have to find the best students you can, and you have to mentor those students. Every lab is different. Every PI is different. Every student is different. So, which is the most wonderful thing about the job, if you ask me. Every day is going to be something new, and either jump in and enjoy the ride, or you're in the wrong business. Is my attitude. So, well, I think one of the, and again, everyone here is much more junior, right? So there's a transition we all go through. The transition is when you're a postdoc or whatever, you're doing your own research. You start your own lab. You're still in the lab doing everything, right? But there comes a transition where you have to hand off that laboratory work and responsibility to your students. And then you have to trust in your students. And that's a very difficult transition for a lot of people. And some people can't make that transition, because if you don't, you're going to drive yourself crazy, right? And so that was one of the hardest things I can remember, is the stress of, okay, I have my 3 or 5 students back then, whatever it is, right? Okay, you know, I think I've trained them well, I know what they're doing, but can I be in my office and know when they generate the data and they're doing these things, I don't have to worry. And it's a gradual evolution in your own thinking, your trust, how you've trained people, and then you jump on from there. And either it works or it doesn't. Most of the time it works, but it's still a very difficult transition. And then the other, I would call the other biggest transition, which I refused to make, and I stand today very happy I refused, is when you get to be get a lab that's been active for a while, publishing, and well supported, lots of students coming through. Then you get the Dave, don't you want to be a dean, or don't you want to be a provost, or don't you want to be a university president? And that's a time for everyone to reflect, very personal reflection. You know, what is it that you enjoy doing, and what is it you don't enjoy doing? And even back many moons ago, I just knew what I tell this to my own students, so I'll tell it to everyone. When I'm driving home from Tufts, and it's about a half an hour for me to get to Concord, I can tell immediately what my day was like by the kind of music I put on, right? So if I'm putting on, hard rock or punk, I know I've had a great day with my students talking science. Point is, if you start to do what I call biofeedback, just understand what makes you happy. Easy decision. So I have never even interviewed for a dean's job or a provost, even though you get asked all the time, because I just know I'd be depressed. It's just not in my DNA. But I have colleagues who love it, right? And that's great. So I still love the day-to-day sitting in my office talking to my students, or having technical meetings, or going off and doing something together as a team. Those are the things that drive me every day. And I don't miss any administrative meetings. It's like the last thing I want to do. No one's ever told me that. I think it's a reflection of your own personality. Perhaps, I'm a bit ADD about things. I'm like constantly amazed at what I see out there, intrigued by new ideas, whatever it is. And, my attitude about the lab has always been, if I can raise money to do the research, and if it's an interesting question to me, then we're going to go do it. I'd still say at the core of the lab that's never changed is this whole idea of how nature makes remarkable materials. That's always at the core. Whether that goes directly into biomedical devices today, based on our silk proteins, whether it goes into scaffolding that supports cells and tissue growth to study the brain or the intestine, or whether it's substrates to grow muscle and fat cells to make the next evolution of food. It's all that scaffolding underpins all of that. So I still think there's a core there, but yeah, I love going in different directions. And as long as we can do it well, I think that's okay. So there's a bit of philosophy there, I would say, in terms of making those decisions to pivot into new areas. So, you know, when your lab grows to a certain level, and our lab really grew significantly when those DARPA programs happened. We had a few DARPA programs back in the mid-2000s, you know, and so all of a sudden we had a very large lab. So we had lots of things going on. And one of the things I decided back then, very, very deliberately, is to try and find something new every roughly 5 years to do that I knew nothing about, because it was a way for me to keep fully engaged, and also just to stimulate what we were doing, right? And so that's how we got going on those DARPA things. And then 5 years later, by design, I decided to start studying the human brain. I honestly knew very little about the brain, but I had a wife who studied psychology, and when she would come home from her clinical counseling experiences, it was a horror show. You know, the patient X was getting this drug and wasn't working, so you try drug Z or you try drug D or whatever it was. So it was trial and error, and you look at neurodegenerative diseases, which were mentioned earlier, there are 0 treatments. And the epidemic of Alzheimer's and Parkinson's and TBI, nobody knows anything, right? There are no cures, no treatments that really, really work well. And so that's when I decided that's about 10 years ago we started that, so 5 years after DARPA, I said, okay, we're going to study the human brain. I knew nothing, like I said, so I hired a postdoc. She was trained in neurobiology, and I sat down with her. I said, look, this is what I want to do. Are you game? And she was like, I'm all in. You know, so Min put together, that's where we built we call it our donut model for the human brain. It's literally a silk donut. It looks like a donut, but it's miniature, you know, like 10 millimeter diameter. And we spent about 2 years just figuring out the right scaffold design to get the neurons to grow well and so on. Anyway, I don't want to go off on those tangents. The point is, you know, from there we've contributed hopefully to things like understanding Alzheimer's more with our models and Parkinson's, and we're continue to do all that. And more recently, we've published a series of papers on viral infections of the brain, which I think are going to be really important going forward because of COVID, and how that leads downstream to accelerated Alzheimer's and things. So that was that 5-year plan, and then the next one was the food. And so by design, and I knew nothing about food other than I love to eat it, right? So, so I find it keeps me very engaged, keeps me fresh, keeps me always wanting to read and learn more. But you have to have that inquisitive nature. It doesn't work otherwise, right? And we still do our other stuff, and some things fade away. So some things get old, and I don't mean old in a bad way, but you know, I think we've made just about every silk medical device we can make. So we don't spend a lot more time on that. We've tried to contribute to the field of collagen biosynthesis, because I still think there's a huge need to develop, you know, synbio approaches to collagen, but we've mostly failed there, so we don't really work on that anymore. So, you know, you try things, they fail enough, you finally give up unless there's a student who's more stubborn than you and wants to keep doing it. So we balance that. Things that just aren't working productively or no one seems to want to fund, eventually you just let them go away, and then we consciously find new things to do. So I can tell you what's next, besides food. I had a brilliant postdoc, Nick, who's back in Canada now. He's running his own lab, and we're still writing proposals together. So this is still the brain, but the next level, because I know nothing about it, is memory, and how does memory work, right? And I know it's a bit esoteric, but the question is, if you read the literature, no one knows how memory works, right? All you find is, and I need to understand at the molecular level, but there is nothing at the molecular level, right? All people talk about is you build synapses that don't exist before, and that's where memory is stored. But that's just very unsatisfying to me. So we've been putting in proposals, using our brain model, and how can you use that brain model? And we've published a little bit on this already to start to track, if you can understand where memory sits and how you can use that in a good way to help people, right? And it begs a lot of ethical questions, clearly, so those are things we'll have to get to. But the idea is as simple as, if I have my mini brain donut model in a dish in a lab, can I train that model to do something it's not normally going to do? And the answer is yes. We've published that you can train the model. People have shown this with neurons in a dish before. We've shown it in 3D. And then the question is, I mean, a bit out there, but if you could train the model in a dish and I implant that into a rodent, does that rodent keep that memory, right? So think about the challenges there, which is why I love these things, right? It's something completely different. How do you measure what the rodent really knows from that donut and all the things that go with that? So that's the next phase, my next 5-year plan, which is starting now with proposals. And Nick's on board, because this is what Nick does. He's just this very creative guy who loves understanding these things. So that's how we do it. That's how we go into new areas by design. And I purposely pick things I know nothing about, so I have to learn along the way. And you just find really good students who really care, and they carry the load. They're the catalyst, like I said early on. And we have to raise money to do it, of course, but you know, we've been very, very fortunate over the years to be able to support what we want to do. So as long as we can do that, we'll keep it going.